Hello, everyone, and welcome to the LabRoots Precision Medicine virtual event. My name is Guannan Wang, and I'm a Senior Research Investigator here at the University of Pennsylvania. And I will be presenting today on bringing precision medicine to veterinary oncology. Please feel free to submit questions during my presentation, and I will follow up with you via email. So first, I'd like to thank Twist Bioscience to invite me here today to share my work and LabRoots for coordinating. I'll share with you our effort at Penn to bring precision medicine to veterinary oncology. So here's my disclosure. On the menu today, at the beginning, I will show you the concept of precision medicine and how it is changing the landscape of human oncology, although I don't think that's a big issue for this audience. Then I'll share with you our study in canine Hemangiosarcoma, applying the concept of precision medicine and the NGS-based diagnostics to identify clinically actionable markers. Lastly, I'll talk about our efforts to build a broad canine cancer panel designed to use in many types of common canine cancers. So I'd like to start today's talk with the story of a 7-year-old German Shepherd dog, Jacob. Jacob was the happiest dog in the world. One day, he started to show lethargy and weakness before he completely collapsed a week after. Ultrasound revealed hemoabdomen and a large splenic mass, which was later diagnosed as hemangiosarcoma. Jacob underwent splenectomy right away to remove primary tumor, followed by chemotherapy to slow metastatic spread. However, despite that, the metastatic disease rapidly appears, and he died 3 months after initial diagnosis. This is one of the sad stories of millions of dogs diagnosed with hemangiosarcoma and rapidly succumb to the disease, and very little can we do at this point to help them. So being a hardcore dog lovers who have witnessed the rise of precision medicine and how it revolutionizes cancer care in human patients, as shown here, by delivering the most effective treatment based on deep patient genotyping and phenotyping. So me and my team at UPenn have the same goal to bring this concept into veterinary medicine. With the help of 2 great veterinary physician scientists at Penn Vet, we set to test the idea of precision medicine in the cancer that Jacob and so many other dogs suffered from, hemangiosarcoma. Canine hemangiosarcoma, short for HSA, is an aggressive malignant sarcoma, one of the most common cancers in dogs, and almost 100% fatal. Most dogs die within 6 months of diagnosis due to metastatic spread to the lungs, and it is overrepresented in German Shepherd, Golden Retrievers, and Labrador Retrievers. These features make HSA an excellent disease to pioneer precision medicine, such that there is a readily available tissue, desperate clinical need, and a potential human clinical correlate with human angiosarcoma, as they are very similar histopathologically, but human angiosarcoma is just too rare to study. We hypothesize that if we assess tumor mutation profiles via next-generation sequencing, we will identify oncogenic drivers and the clinical actionable targets, which will hopefully advance therapy and improve outcome for this desperate disease. To address our hypothesis, first, we performed whole exome sequencing to investigate mutation landscape of canine HSA and identified oncogenic drivers and biomarkers that have potentially prognostic and therapeutic values. Next, I built a targeted resequencing panel to validate these biomarkers in a larger cohort. Also explore the research and the clinical utility of panel sequencing in veterinary medicine. Finally, we'll evaluate these markers in prospective studies, such as clinical trials. Our whole exome workflow is fairly straightforward, except that we have to use canine exome capture solution instead of human, and we did sequence both tumor and the matched normal tissue as a control. The annotation and interpretation of the results, however, is very challenging since canine cancer annotation is very limited at this point or not available at all. So we had to rely heavily on human data to predict and understand the function of the mutated gene, the pathways involved, and most important of all, the implication of mutations in cancer. Our customer canine breast informatics pipeline enable us to identify potential drivers and their biological and clinical implications. Together, our exome sequencing identified candidate driver genes in more than half the cases. Each column here represents an individual case. Case ID shown on top. Each row represents a candidate driver gene. Green squares here show the non-synonymous mutations, and black squares are truncating mutations. Out of the 4 potential driver genes, 2 of them are oncogenes, PIK3CA and the PLC gamma 1, and 2 of them are tumor suppressor genes, P53 and P10. Several points I like to emphasize here. Not only are the genes well-established cancer genes, the mutations we identified here are also well-documented oncogenic mutations as shown in human cancer, such as PIK3CA H1047R, which is the most common activating mutation in PIK3CA gene, and the P10 loss of function mutations, and the PLC gamma 1 activating mutation. So we're very excited about these results. However, we can't ignore the fact that exome sequencing was unable to detect drivers in almost another half cases. Based on several lines of evidence, I hypothesized it's because exome sequencing is just not sensitive enough due to its limited coverage. So we decided to test a more sensitive strategy, targeted panel resequencing, which focuses on a limited number of genes that are involved in hemangiosarcoma tumorigenesis. It's designed based on our own exome sequencing data and, of course, available genomic data on angiosarcoma in both species. It covers a full coding region of 24 genes and hotspot mutation in another 7, so total 31 genes. These include oncogenic driver genes, recurrent mutated genes, and hotspot mutations that are indicated in both species. And I also include a few common cancer genes in human and canine solid tumors, just to widen the utility of this panel. And for method, I chose to use the Illumina amplicon-based targeted resequencing solution. It's perfect for a panel this size and also compatible to use with the FFPE samples because that's what we mostly get. So panel sequencing reproduced exome sequencing findings by re-identifying candidate driver genes in these 4 genes and in the original cases as well. But it also makes new discoveries. It detected potential driver genes in another 8 tumors, as indicated by the triangles here. In these new cases, not only do we see the original 4 cancer genes, I also found new recurrent NRAS mutations in 3 cases. The allele frequency of the recurrent NRAS alterations is relatively low, which may explain why we missed it from exome sequencing data. So the recurrent NRAS mutation we found here is a Q61-R mutation. In human, this is a well-known activating mutation of NRAS and a driver in multiple cancer types such as melanoma and lung cancer. NRAS is an attractive therapeutic target with drugs available to inhibit it. And NRAS is also found, the same mutation is also found in human angiosarcoma, which is an evidence that canine hemangiosarcoma have strong human correlate, could be a good model for human angiosarcoma. NRAS mutant was missed by the discovery sequencing while detected by panel sequencing, highlighting the sensitivity of targeted panel sequencing in this case. So next, we employed the HSA panel described above to sequence additional 30 HSA tumors. Results of all 50 cases are summarized here. I observed the same set of oncogenic driver genes previously described throughout all samples. As you can see, NRAS is observed in 12 out of 50 cases, and 24 tumors bear PIK3CA mutations. The reason I put PIK3CA and PTEN together in the same box is because PTEN and PIK3CA are both key components of PI3K pathway, and PTEN inactivating mutation operates in parallel with PIK3CA activating mutation. Both leads to elevated PI3K signaling and present a very attractive therapeutic target for drug intervention. Out of the 50 cases we sequenced so far, I didn't find obvious drivers in 4 cases. And as you can see, 6 cases have only P53 mutations, while no other major oncogenic drivers. This could reflect the presence of somatic aberrations not detectable by the sequencing method we employed, such as genomic large-scale rearrangement and copy number variations, or could be due to poor DNA quality in these samples, unable to make satisfying amplicon-based sequencing libraries. But overall, the panel is informative for 92% of patients, demonstrating high diagnostic utility. Noted that there is no overlapping between the major driver groups, suggesting that these drivers might represent distinct molecular subtypes of the HSA that were previously thought as a single entity disease, as shown here by this histology image. Several subtypes such as NRAS, P53, and PLC gamma 1, they are found identical. We found the identical mutations between canine hemangiosarcoma and human angiosarcoma, suggesting that although genetically heterogeneous, a subset of canine HSA share genetic similarities between human AS and could serve as a suitable model. On the other hand, PIK3CA, PTEN mutant group, and NRAS mutant group represent 2 great therapeutic targets suitable for pharmacological intervention, and they account for more than 70% of the HSA cases. There are both FDA-approved drugs and trial drugs available to inhibit these targets. The genomic data is very compelling, but the question remains, does the genotype correlate with phenotype? Whether upstream mutations actually lead to deregulated downstream signaling. To that end, we did immunohistochemistry on the same slide, on the same cases. As you can see here, the IHC staining show beautiful signal of phosphor ERK in NRAS mutant cases. Phosphor ERK is a classic marker of a MAP kinase pathway activation, indicating strong association of NRAS mutation with MAP kinase signaling. Also, a direct evidence that NRAS mutant is driving hemangiosarcoma tumorigenesis. Similarly, the activation of PIK3CA signaling is confirmed by the expression of phospho-AKT and phospho-S6, two downstream effectors of PI3K and mTOR pathway. The function study is particularly gratifying since it's consistent with previous findings that PI3K and MAP kinase pathways are frequently activated in canine hemangiosarcoma. And now our mutation data provide a genetic mechanism for these phenotypes. At the end of this process, we have in hand a validated, functional relevant, cost-effective NGS test, which allows us to rapidly screen archived cases for research purposes, but also have the potential to screen standard veterinary patient samples in routine clinical care. This panel allows us to begin to study aspects of canine hemangiosarcoma that were previously unthinkable. In the interest of time, I'll show you here how this panel or genomic findings have powered the design and the development of precision clinical trials, providing not one but two options. In one scenario, phenotyping using HSA panel presents druggable targets. Based on the data I have so far, 50% of HSA cases with PIK3CA PTEN mutations are eligible for PI3K inhibitor or mTOR inhibitor trial, and 25% patients harboring NRAS mutant can be enrolled in MAP inhibitor trial, and there are drugs available. As shown here, these are all the FDA-approved drugs. And but finding affordable drugs is another story. Luckily, our work presents an alternative opportunity by taking advantage of the extremely constrained mutation landscapes of HSA and design listeria vector-based new antigen immunotherapy trial. This study is currently underway in collaboration with Bioscience. And here, as a result of the mutation profiling, we were able to introduce the HSA-specific mutations to the listeria platform and make an off-the-shelf vaccine product to treat HSA dogs that carry these mutations. And hopefully, the specialized vaccine will help train the immune system to recognize and kill cancer cells in a very targeted manner. In either scenario, it's reasonable to think that our genomic approach may lead to novel therapeutic options and transform how canine hemangiosarcoma is treated. This is just one example of the various advancements our genomic approach and HSA panel will make, and a perfect segue to move into the last section of my talk. Here I want to re-emphasize the long-term goal of our collaborative effort is to bring precision medicine to the practice of veterinary oncology. Built on the rich experience gathered in hemangiosarcoma study, I have turned my effort to build a comprehensive canine cancer panel to enable the same kind of deep genotyping, phenotyping, and hopefully a clinical advancement for all canine cancers as we did hemangiosarcoma. Together, our broad cancer panel detects a wide range of mutations in 284 genes known to drive cancer development. It covers a total target region of 1.4 megabases in canine genome, and this is almost 4% of the whole exome. And remember, these genes are all cancer-related. For method, I chose to use the hybrid capture-based enrichment sequencing solution, and this is done by Twist. The goal is to map mutation landscape, identify drivers, biomarkers, to arm researchers and the veterinary clinicians with better prognostic information, targeted therapeutic options, and biomarkers to predict treatment response for common cancers in dogs. The broad canine cancer panel covers genes in key signaling pathways known to play a role in cancer development, such as PI3K, Notch, etc. And the design of the broader cancer panel is multifold. Not only is it targeting the cancer pathways, it also covers potential driver genes in top 10 common canine cancers, such as lymphoma, osteosarcoma, hemangiosarcoma. To the best of our knowledge, this is by far the largest and most comprehensive cancer gene sequencing panel in veterinary medicine, and we are very excited about it. With every opportunity comes with a decent amount of challenges. Here, I want to show you a few lessons we've learned during this process. First, the design of the probes. Most design tools that are currently available are for a human. You would think it's an easy switch between human genome to canine genome. Turns out it actually requires Twist to do some serious coding changes and informatics resources. Eventually, Twist had to basically rebuild the whole design pipeline for non-human species, including canine and mouse. Now that they have it, hopefully future designs for non-human species will be very straightforward. Just a heads up for those of you who are doing non-human genomics. Second, it's a common practice to add blocking reagent to the hybridization reactions to reduce, now the purpose is to reduce non-specific findings associated with repetitive sequences in the genome to improve capture specificity and accuracy. Human COT1 DNA is such a blocking reagent. It is enriched for repetitive non-coding elements commonly found in human genomic DNA. At first, we just assumed that we can use human COT1 in our canine hybridization reactions, assuming that it will do the same with canine samples, but our on-target rate really suffered. Before I did some digging and realized that there is actually not sufficient sequencing homology between human and the canine genome for that to work. So moving forward, canine-specific blockers are the way to go. This is a lesson for us and hopefully for other researchers as well. Currently, the design and the synthesis is all done, and the validation is underway. The matrix looks actually really good based on initial testings with high uniformity and low duplication rates. And we're just working out a few kinks to improve on target. I'm using Hemangiosarcoma cohort as validation sample sets, of course, because their mutation contents are well studied now at this point. I'm glad to find out that the new panel confirms previous results. All the variants that I found that are supposed to be there are picked up by the new panel, such as P53, P10, P3CA, and we also make new detections, such as for like the potential drivers in samples that I didn't find anything before, or I found P53 mutation only. So, why is that? I think combining with the pre-analytical factors for these samples, which mostly have really poor DNA quality to begin with, we think it likely reflects the fact that hybridization approach is more tolerable for poor DNA quality than the amplicon approach. By far, between the discovery sequencing, the small focus panel, and the broad panel, I have almost 100% diagnostic utility for canine hemangiosarcoma. And this is the model I'd like to introduce to other canine cancers and hopefully other species as well. So in conclusion of our effort in bringing precision medicine to canine hemangiosarcoma, our genomic approach allowed us to identify driver mutations in most HSA cases tested, which leads to better understanding of the biology and the pathogenesis of canine hemangiosarcoma as well as human angiosarcoma. We demonstrate that canine hemangiosarcoma consists of multiple molecular subtypes. Some subgroups bear similar driver mutations with human AS, suggesting strong human correlate. And the druggable molecular subtypes, on the other hand, help to guide precision clinical trial design and a rapid evaluation of novel therapy strategy, and eventually tailored effective treatment. Our HSA project serves as a proof of concept study to enable precision medicine in other canine cancers. It already led to the development of the Comprehensive Cancer Panel for canine. Our work here is not only valuable for veterinary patients, we may also be able to translate canine results for similar human diseases, particularly in the case of rare diseases that are difficult to study, to mount a meaningful clinical trial, not to mention finding a targeted therapy. In the end, this is really a win-win situation for both people and our companion animals. So this is my, so this is my funding sources, and these are the people that are closely involved in this effort. And special thanks to Dr. David Ross, Nicola Mason, and Amy Duom. But everyone can't do this without them. And with that, I'd like to close today's talk. And by saying that I'm currently actively seeking collaborations and the tumor samples. Feel free to reach out if you are interested in certain canine cancers or have access to tumor samples. Thank you so much.